Well, hello, and we are back. The topic that we have now is not a typical organ system topic. Going through the organ systems in order has left us in a position where only one is left, and that is the reproductive system. But just like the chapter before urine, when we did metabolism, we focused on the chemical reactions of energy, the provision of building blocks like amino acids and monosaccharides and fatty acids to our body. That wasn't really an organ system chapter, and this one isn't either. But it does truly reflect the experience of the anatomists and physiologists as they began to meaningfully explore the human body. Because in dissecting a human cadaver, you did discover a very repeatable and recognizable pattern of tissues organized into organs. So this is how we got the naming, the anatomy, for the solid contents, the tissue contents of the body. But in addition to the tissues, we noticed that there was a characteristic fluid associated with each chamber in the body. So blood was quickly recognized that you have the cellular component, the hematocrit, being the red blood cell and white blood cell and platelet content, and the plasma, which was a fluid of the body. When you looked inside the heart, you found that fluid, recognized the heart's role in pumping blood. When you examined other chambers, so for example, the stomach had this fluid that was present that was high in mucus, it was high in hydrogen ion, so very low pH. You find bile in the gallbladder and quickly determined that it was produced by the kidneys. And since each of these fluids that I've named and many others are essential to our life, people began to look at them collectively. Water balance, fluid, electrolyte, and acid-base relationships. These are the conditions that actually permit the chemical reactions that we call metabolism. So it's useful to take a somewhat comprehensive look at them and look for them as characteristics that affect the whole physiology, because there are some general effects. I kind of want to point something out. If you imagine you have a sample, let's say you have 100 grams of sand. Now, you know that sand is, each grain is an independent particle. It packs together. Let's say you pour it into a small bottle and start adding water. And the water comes up, let's say you stop slightly above the level of the sand. With respect to the study of fluids, recognize that there's kind of two components there. If you think about the sand grains being hollow and containing a fluid based on water in the cell, its cytoplasm, those water-based contents of the individual grains or the individual cells would be what we'd call intracellular fluid. And because of the semi-permeable membrane, that intracellular fluid, or ICF, based on the cytoplasm and the soluble part of that, the cytosol. The ICF really represents a large number of independent little pools of water solution. So there are very strict control of exchange between the inside of the cell and the surrounding water. And so ICF to ECF, the extracellular fluid, is controlled by the semi-permeable membrane, by the protein carriers that are embedded, by the availability of ATP to power things like the sodium-potassium pump. But the cytosol, collectively in our body, those trillions of cells, each one contains a tiny sample of fluid, the cytosol. that collectively represents the intracellular fluid of the body. This is the living metabolism part, because the living unit is the cell. Now, surrounding the outside of those cells, there is a fluid that's very different in nature. It's called the extracellular fluid. So the first thing we think about is the fluid in the spaces, the so-called interstitial fluid, the cell fluid between the cells. But there are a couple of other pockets of fluid in our body that deserve separate recognition. So, for example, the blood plasma is a circulating fluid being driven by that heart pump that's moving around the body at a great rate, carrying supplies out to cells, picking up wastes from cells. But it's mobile, and it's essentially in interaction through the capillary walls, in interaction with the interstitial fluid in the tissues and in the lungs. But basically, you can look at it and study its properties as a separate body of fluid. All the cellular operations depend in one way or another on water, that polar liquid. We keep coming back to the basic chemistry of water, this polar molecule, and establishes a relationship to all chemicals. Hydrophilic, if water's solutions are easily formed; hydrophobic, if they are not. So how do we study the fluids of the body? The balance of our fluids on an organism level is basically a balance between how much water we gain, the principle of that being the water we imbibe, what we drink through our digestive tract, and the balance of that with the water lost to the environment. There are several sources of this. Every time we breathe out, water vapor that is evaporated off of the moist surfaces of our alveoli are lost. Insensible perspiration is constantly leaving at a low rate through our skin. Insensible perspiration leaves when we exercise. The more the exercise, the more water we lose to sweat. Now, a major water transfer is the water we lose by urination. And finally, although our feces are a semi-paste and our anatomy has evolved to handle it in semi-paste form, it is largely, we consider it contains all our solid waste in a healthy person. However, it's best handled in that kind of semi-paste or semi-solid consistency. That's the condition under which it is easily moved by the muscles of the lower digestive tract. Now, the primary source of gains is the digestive system, but there is a small amount of water that is measurable that we get from metabolic activity, and we go here back to that basic equation from the metabolism chapter. Glucose plus oxygen produces carbon dioxide and water. Water is a product of our energy production. And so this metabolic water is maybe average person 300 mils or 0.3 of a liter per day. Again, a small amount, but not negligible. The primary route of water loss and of water regulation is the urinary system. Let's take a look at these. When we deal with water exchange, we have to balance our nutrients, our ions, all of our essential molecules. Now, when I say ions, I'm talking about sodium and chloride first, but also calcium, magnesium. I'm talking about other cofactors that are needed in small amounts and are required. For example, the micro metabolites are needed in incredibly small amounts. So, for example, iodine, not something that we encounter in the environment at very high levels. But it turns out the hormones of the thyroid gland are iodo hormone. So if you give somebody radioactive iodine, let it pump around the body for a reasonable period. So there's time for that iodine to be taken up where it is used. And then you take the right kind of x-ray. The throat lights up like a light bulb, showing you that basically all that radioactive iodine has been sucked right in to that thyroid gland. So electrolytes follow that same rule as the water, the balance between nutritional intake and loss to excretion. Electrolytes, those charged ions that are highly hydrophilic, are interesting because they give an electrical nature to the solutions that they occupy. Electrolytes, because they are able to transfer electrical energy. So we can, these electrolytes are solutions that can conduct electrical current. We basically are going to balance rates of gain from the digestive system, again, with rates of loss from the kidneys and sweat glands. Hydrogen is a very reactive ion, and as a result, we have a special scale. We track the pH, and you kind of, in chemistry, if you go back and look at your notes, how often in chemistry lab, when you were doing a synthesis, did you have to change the pH? So you got more product if you acidify the solution. In some cases, if you make it basic. The pH balance is very important to reactivity and metabolism. We generate acids in several ways, with the leading generator being the fact that we're making so much CO2, and that water solution generates carbonic acid and bicarbonate with cooperation from a number of enzymes and cells in the body. So we're going to have to monitor and regulate that acid-base balance. And with special attention, what are the acid conditions we need inside the cytoplasm, and what are the acid conditions that we need outside the cells? Urinary and respiratory regulation have already been called out as the main places where hydrogen ion is monitored and regulated. Now, between there, realize the cardiovascular system is the connector between all of the tissues of the body and the outside. So if we're really talking about getting rid of hydrogen ion, you're going to have to carry that hydrogen ion to the lungs to breathe it out as CO2 and eliminate it in the fluids. And you're going to have to transport it using the cardiovascular system to the kidney in order to gain from this acids, reabsorption, acid secretion possibility that happens there. We also noted that a main way we tolerate the acid while we distribute it around the body is through the carbonic anhydrase that's in the red blood cells. So the carrier system, cardiovascular system, is participating in securing that hydrogen ion, converting it into a tolerable form, and basically through transport involving different kinds of binding. We've talked about the hemoglobin, we've talked about bicarbonate, we've talked about that CO2 that's just directly dissolved in the blood plasma, transporting it to a place like the urinary system or respiratory system where we can actually absorb it as through a chemical reaction, producing water, or where we can, in the urinary system, we can directly expel it. When we look at the body, we talk about these different fluid compartments, and there's an old style term, the humors of the body, and that meant the fluids of the body. They recognized that these different compartments each had their own unique fluid. We've encountered this term once before when we talked about humoral immunity, antibody-mediated immunity of the B cells. Now, releasing the antibodies into the fluids produces this immunity, and that's the derivative of this term, the humors. Water accounts for between 50 and 60% of our body weight, and by that I mean not hydration forms of molecules, but strictly H2O molecules. There's a little bit of difference based on gender, with males having a little more water and females having a little less. Most of the water is in the intracellular fluid, contained in the cytosol and maintaining the right connections and the right concentrations for our metabolism. That means that across that semi-permeable membrane, there is a constant potential for transfer of water. And with the transfer of water, you're really changing the concentrations of ions, of metabolites, of nutrients inside and outside the cells. The forces that are at work across that membrane, osmosis, that special kind of diffusion that moves water across the membrane, diffusion, the general force that would work on any molecule, and carrier-mediated transport, whether it's passive, facilitated transport requiring no energy, or whether it's active and you're burning ATP. I don't know if you've made this connection, but when you think about the concentration and diffusion issue, so if you have more molecules over here in my left hand and fewer over here in my right hand, in a space, diffusion is going to move it from high concentration to low concentration. When you move it across a membrane, if you have the right carrier, diffusion would always move it from high to low. So this is sort of what we encountered in the cardiovascular system, in the respiratory system, in the digestive system. There's a flow of a molecule in, the so-called source. And once it crosses membranes or organ walls, it is carried away to another place or consumed, the so-called sink. So diffusion is always going to be moving those molecules in a certain direction, and it doesn't usually take energy. Diffusion is enough on the scale of the single cell. It's a powerful force on that scale. But what happens when you want to accomplish the opposite? Meaning that you have a high concentration on one side and a low concentration on the other, and you want to move the ion from the low concentration to the high concentration. That's what's going to take ATP. That's what's going to require active transport. We saw this with the sodium-potassium pump in the nervous system. If you recall, the sodium-potassium pump in every membrane is pumping sodium out and potassium in to the cell. 1 ATP will move 3 sodium out and 2 potassium in. Now, the ATP is the energy needed to pump against the concentration gradient. In other words, to overcome diffusion. So those basic processes that we keep returning to, because they explain what's happening between these compartments that we call the extracellular fluid and the intracellular fluid. I mentioned the intracellular fluid is the cytosol. In our body, trillions of tiny compartments that are separated from one another. But I didn't really complete that story by talking about the extracellular fluid. You think about if I'm outside any cell in the body, so I'm out here, there's another cell right here, and the space between is filled with extracellular fluid. But if you kind of take a cruise in that extracellular fluid, you can go around this cell to a space between these other cells. So the extracellular fluid might consist of tiny spaces, but it basically is a continuous body of water because it's surrounding the outside of the cellular membrane, the plasma membrane. So if you had the right dimension and you've got a good imagination, you can imagine getting in your submarine and basically cruising around the outside of each of those cells in the extracellular fluid. So this includes the major subdivisions of extracellular fluid. The interstitial fluid is the fluid between the cells that are densely packed together. And the second, though, is the plasma of the circulating blood, that liquid medium that suspends the blood cells. And through pumping that plasma, we push those blood cells with their various functions around to all living cells of the body. Now, the intracellular fluid, as you might imagine, is different. It has many more subdivisions, and here's why. We started out life as a single squishy cell. Cell division produces a lot more cells, but they quickly differentiate into the specific tissue types and combinations of tissues that make our different organs and our different organ systems. So there's only one place in the body we're going to make a stomach, and that's the only place that we're going to make hydrochloric acid in big volumes. So that's going to be a characteristic of that fluid compartment, and it's an extracellular fluid that surrounds the cells. In the lymph system, we have seen lymph as a general means of returning water to the bloodstream because of that unequal extraction and return around every capillary in the body. The perilymph and endolymph are lymph fluids that are present in the inner ear. Cerebrospinal fluid in the compartments within the meninges. So the spinal cord and brain, that whole structure, the central nervous system, is wrapped in meninges and managed by central spinal fluid. Now, it's not pumped around and back and around and back. It's basically secreted by those specific glandular structures up in the ventricles of the brain, and it is the pressure of the production that pushes the older spinal fluid ahead of it, and eventually it circulates around to all the surfaces and basically leaves through those granulations that penetrate to the arachnoid mater, and from that point, after it's circulated around the central nervous system tissues, it's picked up waste, it's picked up contaminants, and that exiting cerebrospinal fluid is treated as waste by the body. Synovial fluid is present around each of our highly mobile joints, our synovial joints. And that synovial fluid is characteristic. It adds hydraulic support to each of our joints. Serous fluids are those fluids between the layers of our serous membrane. You remember those are always double membranes. So pleural would be a fluid around the lungs, pericardial around the heart, and peritoneal is that large abdominal membrane, the greater omentum is part of that, that suspends our digestive tract in large part, but also several other associated organs. The aqueous humor of the eye is another extracellular fluid. And what I want to mention is that for these extracellular fluids, each one of their ionic and molecular content is critical to their function. I don't know if you know this, but certain disease conditions can change the content of the aqueous humor in the eye in a way that blurs the vision. I am familiar with a case where a diabetic attack, the blood sugar went high and stayed high long enough that sugar actually diffused into the aqueous humor in the person who experienced this. Out of control, type 2 diabetic, woke up and was legally blind. He said that if you had taken goggles and smeared them with Vaseline, that's what her vision looked like. In other words, if someone was over here wearing a red blouse, she would see a blob of red that was very fuzzy, but she couldn't identify it as a blouse or as a person. Now, happily, she went back on her meds, stopped eating whole batches of fudge, and in time her vision returned as the contents of the aqueous humor returned to what was normal for her. Now, but among these subdivisions, we're going to have exchange, and the cardiovascular system is going to be the big linking agent. Remember, if we think about a block of tissue, here's the capillary bed we're shoving blood through there to supply oxygen and nutrients and pick up wastes to produce lymph and have it be conducted off by the lymph system. So this is just a reiteration. This is occurring around capillary beds, and of course capillary beds penetrate each of these extracellular regions. The interstitial spaces to the plasma of the blood for their main transport. And of course, every time you have capillaries, you have the lymph system draining the excess water that is lost around those capillaries. So we've simplified the human body and the carbon molecules. How does our solid composition correspond to these different fluids? Over on the left, we see the solid components, those identifiable categories of molecules that exist within these solutions. And we notice we're basically just reminding ourselves of a couple of things here. The most abundant and the most diverse type of organic molecule is the proteins. Sometimes proteins are made in massive amounts, like the actin and myosin of muscles or the hemoglobin of blood. Sometimes they are individual enzymes that are only active in a specific group of cells in a specific organ in the body, but they add up to a tremendous amount, 15 kilograms of solid. Close behind them are the lipids. Lipids are incredibly important as membranes, as components of membranes, producing not just the compartment we call the cell, but all the compartments we call organelles. Those membranes and their differential selective permeability is going to control and regulate the appropriate metabolism. And right behind it, about 10 kilograms, is the lipid component. Now look at this. Minerals, those things that form electrolytes. So here we call to mind our electrolytes in the blood plasma: sodium, potassium, chloride, calcium, magnesium, for proper control and supply and proper transport. But also we would ask the question, when we're making something like bone, soluble components are delivered by the blood plasma. But when you turn cartilage to bone in endochondral ossification, what's actually going on? That's flowing in the blood plasma, not just calcium, but the ionic and molecular components like carbonate and phosphate. Phosphorus being a very important mineral that has to be delivered in order to make phosphate. So minerals, actually about 3 kilograms, exceed the amount of carbohydrate in the body. Carbohydrate is wonderful as quick energy, and because it's so sweet, we notice it. You'll notice it doesn't compare to proteins or lipids in its total content. Finally, we have all the rest, and they're lumped together here in miscellaneous, meaning that, if you look, maybe if you're generous, 1 kilogram represents the huge mixture of other molecular types that are present. Notably, and I think important, we named a big 4 for the carbon polymers of the body. They were proteins, lipids, carbohydrates, and nucleic acids. There's not enough nucleic acid to actually measure it on a kilogram basis for this comparison. It's included down here in miscellaneous. Now, with respect to water, we have most of the water, over 20 liters, is the intracellular fluid. For intracellular fluid, think ICF, think cytosol, and you're done. The water base of that cytosol is well established, even though the cytoplasm itself contains lots of organelles and a gel-like consistency. Over here, the extracellular fluid, notice the interstitial fluid, the fluid that's continuous all over the body surrounding all the cells, is the major point. We can identify the plasma as a measurable amount. And in this case, it looks like what? 2 liters, 3 liters, something like that. And finally, all of the other interstitial fluid, extracellular fluid, is lumped into a category and a relatively small volume. As you can imagine, in the cochlea and the semicircular canals, in the auditory meatus of the temporal bone. Inside that cochlea, there are lymphs that affect our hearing. They're not very large in volume. They're part of that they're part of that other category shown in this slide. Now, comparing males and females, there is a slight difference in water content, and it basically relates to the hormonal effects that relate to reproduction. So, a male with testosterone develops higher muscle mass, and as a result, hydrates that muscle to a higher extent, whereas the female body has a higher content of fat that relates to her role in producing eggs. So we do see this one difference of 40% solids in males and about 60% water. In the solids in female go up to about 50% because the water content is slightly smaller. But basically these components are fairly, other than that one difference of muscle and fat, are fairly comparable. The extracellular fluid contains all of the components that vary regionally depending on the function of the organ or organ system. So we don't find the same content of these subunits in all tissues. Good example, we mentioned earlier when we were talking about nutrition that a special cell type called an adipocyte was the site of fat production. When you put oil into the vacuole of that cell, you basically have a fat cell. So those regions where the adipocytes are and you are storing fat would be areas where the particular lipid content is high compared to proteins, carbohydrates, and other nutrients. Extracellular fluid and intracellular fluid are called compartments because they often are maintained at different levels, and they act as different entities in the metabolism of the cell. The reason for a barrier, a membrane, or on a higher level, the membrane of a cell like a plasmalemma, or on a higher level, the membrane of the lining of a compartment, is to maintain that separation so you can have different chemical, you can contain chemical conditions and have different reactions. We're going to separate this, and we're going to use part of the control mechanism are the carriers and the active transport mechanism that establish these chemical differences. Now, what does that mean in terms of our electrolytes? We find a very distinct difference between extracellular fluid and intracellular fluid that reflects the difference between being outside the cell, where it's basically a non-living bath. The fluid bath that surrounds the cell is the extracellular fluid. And within the cell, the living cell, that intracellular fluid, which is actually conducting the metabolism and keeping the cell alive. So how does that translate to our electrolytes? We find more sodium and more chloride outside the cell than inside the cell, and the sodium-potassium pump has already kind of explained that to us. Bicarbonate is high in the extracellular fluid, and you recall that the various ways of moving carbon dioxide included things like dissolving CO2 in water to a small degree, but much more of it was converted to this carbonic acid and bicarbonate equilibrium, bicarbonate being a form that can be easily transported in the plasma and after it's made in the extracellular fluid. But basically when you make that CO2, you're making it inside the cell, inside the mitochondria, and that CO2 is then exiting. You're basically pumping that CO2 out of the body to maintain a low CO2 concentration and to maintain proper hydrogen ion balance. In the intracellular fluid, those ions that we find are going to be more associated with the metabolic reactions in the cell. So potassium, magnesium, and phosphate are all used on a regular basis from inside the cell. We also find a huge population. We can't give a list here because they are so different from region to region in the body and also so diverse. However, when you take the proteins that are being made from the genes in the body and you put them in the cytoplasm at their normal pH, most of them have a net negative charge. And I don't mean all of them are -1, because proteins are so large, they might be -2, 3, 4, they might have a net charge of -5. And that contributes to the electrical difference that we call the transmembrane potential, that constant presence of proteins in the cytosol. So let's take a look at these in summary form in these graphs. Here is the intracellular fluid, think ICF, think cytosol, and the cations that we find in the cytosol. Very high potassium, that's that sodium-potassium pump. Relatively high levels of magnesium. Now the reason for that, so we haven't made a big deal out of magnesium. Okay, where's all this magnesium coming from all of a sudden? It turns out that many reactions that go on in the cytosol need magnesium as a cofactor, and those reactions are so diverse and so numerous, we don't make a list of them. It's just that it is a general cofactor that goes along with the enzyme that's needed to perform that chemical step. Now notice a small amount of calcium present inside the cell. Now, this small amount, though, is critically important. Calcium is needed. We've seen several places where calcium is stored, like in the sarcoplasmic reticulum or in the fluid around the synapse, within the synapse around the telodendrial junction. Those calcium amounts are necessary to contract a muscle or to fire a nerve. Up here we have sodium. Notice a small amount of sodium inside the cell. Inside the cell, the negatively charged ions, here's those proteins, quite a bit. But here's hypophosphate, lots of phosphate. Now, in a way this makes sense, since energy in the form of nutrients is being transferred to a carrier called ATP. To be sure that this represents a continuous flow, you have to have a ready supply of phosphate. Phosphate is also used in DNA synthesis. So every time you repair DNA, every time you replicate DNA, you're going to need phosphate. Relatively low concentrations of bicarbonate. So we're making it constantly, and then we're transporting it out. Chloride is low, part of the initial observation about chloride, but notice that sulfur is present in almost equal quantity to the chloride and a relatively small concentration. So inside, big thing to remember, potassium and magnesium, phosphate and proteins as major components. What about outside? Well, here we have two different pools. The first we'll look at is the plasma, the circulating fluid carrying the blood cells. What do we find? Lots of sodium, lots of chloride, the main determiner on the ionic side of osmotic pressure. Here's that bicarbonate being transported out. Relatively low phosphate. Now, phosphate's being transported to cells, but being absorbed by those cells because that's where it is used. Potassium, calcium, and magnesium are shown over here on the left. Relatively low concentrations, but very important. These are the supply levels that are transporting these two. the intracellular fluid. So what do you suppose it means? Go back. If we're going to maintain this level of potassium, and this looks like it to me, it goes from 40 up to let's say 180, 190. 150 milliequivalents of potassium are present in the cytoplasm of the cell, 150. But in the plasma of the blood, we're only seeing about 10. Now, that 10 is accounting for the delivery of the potassium to the cell. What is suppose you're pumping it from a region that has 10 milliequivalents to a region that already has 150? That means you're going to have to use energy. That's that sodium-potassium pump, isn't it? Transporting it in at a certain level, but then concentrating it at a much higher level. You're going to need energy to move that. So lots of sodium, by far the dominant cation. The dominant anion is chloride, with proteins and their negative charge being present in the plasma. Now, most of these are the blood proteins that are circulating within the plasma. But organic acids, sulfates, and bicarbonates also are represented. What about the interstitial fluids? We see the same basic lesson, lots of sodium and lots of chloride. This is the reason we crave salt. Lots of salt to maintain our ionic contents and the help with the osmotic relationships. Now, we're going to find later that with respect to water movement, the cell doesn't have the ability to grab a water molecule and shove it across a membrane. But it does have the ability to pump these ions. And so using these ions, if I pump sodium or chloride across the membrane, the water is going to follow by osmosis. And so it's the pumping of ions that is critically important for maintaining water balance. When we look at these interstitial fluids, now again, I want to emphasize this is a continuous pool of fluid around the cellular entities inside the body. So sodium is the big cation with potassium and calcium being found, but again, only in supply levels. The anions, chloride is the major ion with bicarbonate, phosphate, and sulfate, together with the organic acids and proteins. But notice something here, around the cell, that level of proteins is fairly low. And that's because there isn't a lot of need. There's not a lot of metabolism involving proteins, either as structures or as enzymes that occur outside the plasma membrane. So what do these fluid compartments actually amount to? Things to remember: plasma membranes are selectively permeable. Ions are specifically transported. The channel may be passive, but it would be something so specific, we might call it a slow leak potassium channel. That means only potassium moves through it and always at a slow rate. Carrier mechanisms are those things that are under the control of the body. If it's a source-sink relationship, the fact you're constantly feeding it into one side dictates which way it's going to go, because diffusion is going to move it. But the fine control comes from the ability to use energy to selectively pump things across in one direction or another. Osmotic concentrations are going to be always in balance between the ICF and ECF. Water moves passively. If you have an identical total solute concentration, let me stop right there. When we do something in the chem lab, let's say it says, I want a 10% solution of sodium chloride. Now that expression, not molar, but percent, is by weight. So you weigh out 10 grams, put it in a volumetric flask, put in enough water to dissolve it, and then dilute it up to 100 milliliters. Now that is a measured salt solution. And in our body, we don't ever encounter that kind of purity. What we have is lots of different molecules and ions in solution together. And the first point I want to mention, when we talk about osmotic pressure, we're talking about the total dissolved content, the total solutes. It makes no difference what the molecules are. The more there are, the higher the concentration. And so you can pump, sodium and chloride are available in high concentration. You can pump other ions as well. So if you have a membrane right here in the middle of your picture with two sides, and you're going to have to move water across that membrane. If the total solute concentration on the two sides is the same, we call that isotonic. Water is moving left to right, but it's moving right to left at the same concentration, at the same speed. So those cells would have a stable size. They wouldn't blow up and they wouldn't shrink down because the water is in balance outside and inside. If there are changes to that, let's say there's a huge increase in the salt concentration, that's going to move water toward the higher concentration until the osmotic pressure, the concentration, is the same. So let's take that membrane picture again. We have a lower concentration to start over here of solute and a higher concentration on my right of solute. Now, a lower concentration of solute means a higher concentration of water. And a higher concentration of solute means a lower concentration of water, so the water is going to move that direction. As it moves this direction, the concentration of water here drops, and the concentration of water here rises until they are equal, and then the exchange becomes equal in both directions. This is the way we adjust our water balance. So when we talk about this, it's kind of complicated because in chemistry, we really focus on the volume of the solvent and the molar concentration or the weight concentration of the solute. But here you have to think of the relationship between both. Let's think about how the body's going to regulate water balance. First of all, homeostatic mechanisms will involve receptors that are monitoring conditions. And you may ask the question, how do we keep track of at the same time, the water content and the solute content and the different contributions from the different solutes? It seems like a great big problem. Well, remember back to the basic definition of intracellular fluid and extracellular fluid. Intracellular fluid is trillions of tiny droplets. Extracellular fluid is one big pool. So the body tracks the composition of extracellular fluid because it's much more general. You would have to have separate receptors in each cell to track intracellular fluid. No receptors directly monitor fluid or the electrolyte balance, the different concentrations of the different electrolytes. Cells can't move water molecules. That's something that they basically did not develop. It makes sense, doesn't it? We think cells arose in water, and specifically in salt water. So if they were alive, they were bathed in water all the time. They only had to maintain their cellular concentrations in order to stay alive. So they didn't have or need water transport. And by the way, that basic property of membranes is retained in terrestrial organisms like ourselves. The body's water content is going to rise if you overhydrate or if you hydrate, if you drink water. If you stop drinking and continue your water loss, then there's going to be a net loss of water, and it's going to involve a balancing of those osmotic relations between ICF and I and ECF. Now, these are under various levels of control. Hormones control water balance on a whole body basis. We've talked about ADH produced in the hypothalamus, stored in the posterior pituitary, and released whenever we want to retain water. So the effect of ADH that we've seen so far is to regulate the concentration of urine. It has an effect of concentrating urine. It's associated with aldosterone, which has the same effect, concentrating the urine, minimizing water release. Natriuretic peptides opposed the effect of these two antidiuretic hormones. The effect of natriuretic peptides was to increase water release. So ANP, its effect on the kidney is to produce high volumes of urine that are low concentration. ADH, oh, I'm sorry, I forgot I had spelled this out. ADH, antidiuretic hormone produced in the hypothalamus, stored in the posterior pituitary, a water conservation step at the kidneys. Shows perfectly the effect of an endocrine release. It's released from within the blood-brain barrier and basically taken up by those fenestrated capillaries and those gap capillaries. Rapid transmission all over the body. At the kidneys, its effect is to change the concentration of urine by reclaiming all the water out of the filtrate that you possibly can. Its main effect is on the collecting duct, the collecting tubule, and on the extraction of water in that final trip down through the pyramid to that calyx deposit. Now, osmoreceptors, I'm doing a lecture. I can't stop now. Go ahead and take the trash.
No, we're just good.
Okay. Excuse me for the interruption. That will be something that's historically present in this recording now forever. There are osmoreceptors in the hypothalamus that measure this water movement and osmotic pressure and cause the secretion of ADH. Aldosterone comes from the adrenal cortex of the kidney, shows you that the same kind of impulse, retaining water, can come from different endocrine glands altogether, and they react to something different. Chemoreceptors notice rising potassium, approaching the upper limit or exceeding what we call hyperkalemia, or falling sodium levels in the blood. They activate a cascade. The kidney releases renin, which produces that cascade that I've named before: angiotensinogen, angiotensin 1, angiotensin 2, aldosterone. And basically, it affects the rate of sodium absorption and potassium loss. This is in the distal convoluted tubule, so it's a late nephron effect, and the collecting duct. The thing to remember about osmosis, and I keep drumming this because, frankly, if you extend your knowledge of diffusion, which most people understand, to include a real understanding of osmosis, you've gotten beyond what most general chemistry students can achieve. The thing to remember, the cell can pump the salt. The water follows the salt. By salt, we mean those ionic components that we call solutes that are easily dissolved and transported in the body. And a list of this would be very long. High aldosterone plasma concentration causes the kidneys to take that salt out of the filtrate and put it in the tissues around those tubules. The increase in concentration of salt around the tubules is going to draw more water out of the filtrate and concentrate the urine. Now, what that amounts to is not water addition. You're not hydrating the body, but you're retaining the water that's in the body. And as a result, you're delaying that critical threshold of dehydration where you would start to affect your metabolism. Natriuretic peptides, you'll recall, were simple. They simply countered the effect of ADH and aldosterone and angiotensin, those products of the renin cascade. And they basically are released by the cardiac muscle when there's abnormal stretching of the heart. Now, think about this. If your water content is too high, your blood pressure is rising from the increase in volume of the blood. Now it's stretching the heart more from that increasing volume, and that causes ANP and BNP to be released. It simply reverses the effect that would be present under ADH and aldosterone. It causes release of high volumes of low concentration of urine. So rather than extracting that water, you're pumping it out and filling the bladder faster. And actually, this is something the body can do pretty quickly, is dump excess water. So the relationship we want to establish is summarized here. This intracellular fluid shown by this represented single cell surrounded by its plasma membrane. Now we are noticing an input here because all the cells of the body are producing about 300 mils of water every day. This is what we call that metabolic water, and it joins the water. They're saying here a normal intake would be 2.2 liters. So we're talking about a 2.5 liter day of water input into this extracellular fluid pool. And the losses. Water vapor lost at the skin and the lungs. This is insensible perspiration, and you'll notice almost, well, over half of the water that we lose is that passive loss, that insensible perspiration. The feces are dehydrated to the point where we only lose a minor amount. And the urine is a major amount of the water loss, fully close to half. Now over here, this dotted line, this is an it depends line. So if you exercise heavily and lose an additional liter here to sweat, then you would add an additional liter of input here in order to keep these two chambers in balance. So this is that variable day-to-day situation that's based on your actual physical activity. This overall chart shows you kind of everything, and there's more information here than is labeled. Do you notice each chamber is sized based on the amount of intracellular fluid and extracellular fluid that we find in the whole body? And you'll notice that the level up at the top is the same because the spacing of those dots, those are the solutes. That's kind of graphically supposed to convey to you the concentration of solutes. And when it's roughly the same in the two chambers, remember over here, we have trillions of tiny compartments that are each surrounded by the common fluid of the extracellular fluid. But this is in balance. Now, what happens if you have a water loss that decreases the volume? Maybe this is due to a hemorrhage situation. You bleed out, and so you lose this amount of water. Do you notice how that concentrates the extracellular fluid? That will produce an osmotic movement because the solutes are lower in concentration over here. That means the water is higher on the intracellular side, and the solutes are more packed together here on the extracellular side. That means the water concentration is lower, so osmosis moves things from left to right. Now, if this resulted as a result of pumping solutes over, the same kind of movement would occur. Notice that as you pump water over here, the intracellular fluid volume goes down and the extracellular fluid volume goes up until they're levels until this equilibrium is established, and also returns the spacing of these dots to the same kind of relative position. So you're basically adjusting the ionic concentration with that water movement. Electrolyte balance. Balance is a gain and loss on a daily basis. It's going to affect the water balance and the effect of osmosis. In addition to the ions that we focused on in this chapter, you'll recall back at the blood chapter, we had several general blood proteins: albumin, fibrinogen, globulin. And albumin, especially the most abundant one, was there for the maintenance of overall osmotic balance. The different ions do have different kind of native levels or native concentrations, and we're going to deal with these in detail and look at them specifically, how they affect the overall metabolism of the body. So, for electrolyte balance, the main problems are due to an imbalance of sodium ions because it is so plentiful and so abundant. If the sodium gets out of balance, it produces general effects that are related to the total solute concentration. Now, potassium balance would be second on the list, and it's much less common, but it's more dangerous because when potassium is in the wrong concentration, what you're actually doing is stopping metabolic reactions within the cell. So where osmotic relations are affected, extracellular fluid is affected strongly by sodium. The metabolism inside the cell is affected more strongly by potassium. So let's take a pause here. We've been going at it an hour and 15 minutes. I'm going to stop this show. We're going to take a quick break.